Hello, everyone. Good evening. So, today we will be discussing with Unit 3, Part A and B both, right? So, A is talking about replication, B is about transcription. I hope by this time you all have gone through these topics, we have revised these topics, right? So, let's quickly go through the questions. So, as you can see here, question number 1 is showing you the replication fork. Okay, so this is look at the polarity 5 to 3, and here you have 5 to 3. So this is your obviously the leading strands, and these are your in fragments. So that's mean these are your Okazaki fragments, right? Now, you already must know about the concept of Okazaki fragments, which one comes first, which one comes second, and all. So, focus on that concept and answer the questions here. So, look at the first statement. The first statement is saying that 1 represents the leading strand, while 2, 3, 4 represents Okazaki fragment, which is true. Second says that among the Okazaki fragments, synthesis of 4 occurs prior to synthesis of 3 and 2. Let's understand this. If your leading strand is this side, that means here you have the helicase and the helicase is moving this side. Correct? Now, if your helicase is moving this side, that means it was once present somewhere here. So this section of the DNA got exposed first, followed by this section of the DNA, followed by this section of the DNA. Okay? Now, if this section of the DNA has been produced first, that means this is your first Okazaki fragment. So therefore, statement number B is also correct. All right? So the correct answer is option 3, A and B. Next, you have to match the table. But what they are saying? A researcher wanted to test the effect of different chemical agents on double-stranded DNA. Double-stranded DNA was taken in tubes A, B, C, and D, and 4 different agents were added individually to each tube. The researcher, however, forgot to label them. The properties of the added chemical agents on double-stranded DNA were analyzed. The possible product given in X and the specific action of the chemical agents listed in Y is given, and you have to match the 2 columns. So just read the columns and see if you can match them. This is very simple. So if you want to get only the nucleotides, that means what? Just think of the DNA structure. You have the sugar, you have the base, and the sugar and the next one is attached by the phosphodiester bond, right? Here also again you have the sugar and you have the base. Again you have sugar and the base, like this, right? This side also you have the same thing. You have the sugar, you have the base, and then you have the phosphodiester bond. Okay, so if you just want to get the nucleotides, that means what you have to do, you have to break the phosphodiester bond, right? So to get nucleotides, you have to break phosphodiester bond. If you only want the nitrogenous base, you have to break this bond. What is this bond? It is a glycosidic bond. If you only want to get the nucleoside, that is the sugar and the base, then you have to break what? The phosphate. You have to remove the phosphate groups. And if you want to get only a single stranded DNA, you have to break the hydrogen bonds. So single stranded DNA you will can be obtained by breaking the hydrogen bonds. Okay, so you have to be very thorough with the different bonds which are present in your DNA. All right. Now looking into the third question. Now the third question is saying the long DNA strand depicted below is serving as a template for lagging strand DNA synthesis. The short lines represent the newly synthesized Okazaki fragments. Okay? In which position among A, B, C, and D would DNA primase act next? So, what do you think? Option A, B, C, D. At which position would DNA primase act next? May I get some answers? Give me some answers. Let's see. This is a very simple question. Okay, you're saying D. Others? Okay. Come on. Everyone, give me some answers. We are all learning here, right? This is a learning platform. Doesn't matter if it is right or wrong. We are all learning. So trying is what is important. I keep saying this to everyone. It's more important to try. So the correct answer is D. Why? Because as you can see here, the Okazaki fragments have been synthesized already. Okay. So if it has been synthesized, just check this out. This is 5 to 3. That means this is your 5 to 3. That means this is a place where the primer has been synthesized. Similarly, here, this is a place where the primer has been synthesized. This is the place for primer synthesis. Now, here also a primer will be synthesized and then that will be then elongated. Okay? So, the next primer that will be synthesized will be at the position of D. Okay, so please check the polarity whenever you get these kinds of questions. No, the hint is given to you, the polarity is given to you, so just put the polarities on the other strand and you will be able to understand what or which part is the right one. Now next, so next question is saying which one of the following statements related to transcription and processing of mRNA is incorrect? Minded, they are asking which is incorrect. So, number one, during prokaryotic transcription, DNA binding properties of RNA polymerase are altered by sigma factor. In eukaryotic transcription, synthesis of rRNA, mRNA, and some small RNAs occurs by RNA polymerase 1, 2, and 3 respectively. Splicing observed in tRNA involves successive sequential cleavage and ligation reaction, while pre-mRNA splicing proceeds through lariat formation. And then mRNAs with premature stop codons are degraded by nonsense mediated decay, and mRNAs without in-frame stop codon gets accumulated and translated in the cytoplasm. So even if you don't know what is nonsense mediated decay, doesn't matter. The other things is something which you know. So why don't you start ruling out the other statements? See if they are correct or incorrect. Okay. Sarita is saying option 4. Anyone else would like to try? During prokaryotic transcription, DNA binding properties of RNA polymerase are altered by sigma factor. This is true. Why? Because sigma factor is the one, it is the subunit of your RNA polymerase which recognizes the promoter site, right? So it comes and binds to the promoter site, and then the core RNA polymerase will come and bind to the sigma factor. Okay? Next statement. In eukaryotic transcription, synthesis of rRNA, mRNA, and some small RNAs occur by Pol 1, 2, and 3, which is correct. Pol 1 is for the synthesis of the rRNAs, except for your 5 srRNA, which is synthesized by Pol 3. Pol 2 synthesizes your mRNA and most of the small RNAs, small nuclear RNAs and all. But yes, some of the small RNA, small RNAs are synthesized by Pol 3 also. Okay, so this statement is correct. Next, you have statement 3. Splicing observed in tRNA involves successive sequential cleavage and ligation reaction, while pre-mRNA splicing proceeds through lariat formation. So, as we have seen in tRNA processing, you don't have a proper splicing mechanism. tRNA, rRNA, there is processing happens. Okay, some unwanted nucleotides and all, they are all removed. But not a very established splicing process you will not get. Okay, it is only found in the mRNA. So therefore, statement 3 is also correct. Now, if all the 3 statements are correct, then obviously statement 4 is incorrect. So many a times in your exam, you will get some statements which you might not know. Okay, so in that case, try to eliminate the other statements. Check whether those statements are right or wrong. Okay? Now moving to question number 5. So following statements are made about double-stranded break repair model of homologous recombination. I hope everyone has revised this part because we have taken up replication and repair earlier also. Okay, in between a lot of time has passed, right? And I'm expecting that you must have revised. So what are they saying? Process of double strand break repair recombination is triggered by introducing a double strand break in a DNA duplex. In a process known as 3' end resection, the exonucleases, along with the DNA helix, degrade one strand on either side of the break and generates 3' single-stranded termini. One strand of the donor duplex is displaced due to formation of heteroduplex DNA and generates a displacement D loop. Branch migration allows the point of crossover to move in 5' 3' direction of recipient strand. Completion of DSBR recombination may generate either crossover recombinant or non-recombinant product. So which one of the following combination of statements is correct? Okay, Debashri is saying option 4. Saritha is saying option 2. So the correct option is option 2. Okay, A, C, and E. Okay, that is the correct answer. Next, you have question number 6. The following statements are made with reference to the fact that tRNA are known to possess T in this sequence. Where do you have the T? Where do you have thymine in the sequence? Do you remember? You have the D loop, you have the anticodon loop, you have the T psi loop, right? You have the T psi loop, you have the D loop. Why do you call it as a D loop? Because a uridine has been converted into diuridine, right? T psi loop, why? Because the uridines have been converted to, some of the uridines has been converted to your pseudo uridines, and a particular one has been uh, converted to your thymine. Okay? So, if you look at the statements, they are saying that statement 1, RNA polymerase 3 utilizes TTP as one of the substrates. Like any other RNA polymerase, RNA polymerase 3 also utilizes RUTP, but when the RNA pol 3 reaches a loop structure, it bends to S-adenosyl methionine to methylate C5 position of the incorporated uracil to result in the thymine in the co-transcriptional manner. During transcription of tRNA genes at the designated position, DNA polymerase replaces RNA pol 3 to incorporate T in the tRNA. A specific methyl transferase utilizes a methyl group donor to post-transcriptionally modify the specific uridine residues to T residues. Uracil to thymine conversion occurs in a large number of tRNA in the T psi C loop. Sorry. So, so what do you think can be the answer? Okay, Sarita is saying option 4. Others? The correct answer is option 4. A specific methyl transferase utilizes a methyl group donor to post transcriptionally modify specific uridine residues into thymine. The thymine is incorporated after transcription, not during transcription. Okay, during transcription, RNA polymerase will only be able to incorporate uridine against adenines. Okay, not thymines. So it has been modified after transcription. Uracil to thymine conversion occurs in a large number of tRNA in the T sil loop. That is why, because here you have a thymine, that is why the name has come as T sil loop. So you have thymine, you have the pseudouridine. Okay. All right. Now coming to a very easy question. It's just based on the polymerases. So given below are 4 sentences with blanks. Now I feel this is a very easy question. What about you? So all you have to do is they have given you RNA pol 1 transcribes dash. miRNA genes are transcribed by dash. The RNA polymerase found only in plant is dash. Tasi RNA are synthesized by dash. So this is something, these three are something which we have discussed in class as well, isn't it? RNA pol1, miRNA, RNA polymerase found in plants. So at least those who have attended my class, you should be knowing this. So let's see, what do you say? I only find Sarita answering. What about others? What happened? If you're here, then I would expect you to participate also, isn't it? Otherwise, what's the point of conducting these kinds of discussion? This is a discussion. Come on, give me some answers. There goes Devashri. Okay. Tell me what you're thinking. Again, I'm telling you, I'm not evaluating you here. Whether you're right or wrong, this is not my evaluation. This is your own evaluation. Okay. So you say the option is 3. So this is the correct option. So RNA pol 1 transcribes what? In first case, they have given you mRNA. Wrong. tRNA. Wrong. 45 is rRNA. Yes, this is correct. 18 is rRNA. Yes, this can also be correct. Okay? Two can be correct. Now look at the second sentence. miRNA genes are transcribed by. This is a tricky thing. miRNA genes are present within the nucleus, or more specifically, it is a part of the DNA itself. miRNA. siRNA comes from outside. It doesn't have any particular transcript. Okay? But miRNA has a transcript of itself. So therefore, like the normal DNA which is getting transcribed, the normal genes, not the normal DNA, the normal genes which are getting transcribed by Pol2, your miRNA gene also gets transcribed by Pol2. So here you can make the difference straight away. Okay? Even if you don't know C and D, still you will be able to give the correct answer. Did you notice that? So C is saying that in polymerase found only in plant is pol 5, even pol 4. Okay, 4 and 5 both are found in plants. And task C, RNA are synthesized by pol 2. Okay, so these are some tricks, as I told you, elimination. There are many ways of answering questions. Okay, now question number 8. So I'm almost to the end of the discussion. Like every day, I've come up with 10 questions only. So this is question number 8. Here they are telling you a tree construct has been given to you, right? So researcher wanted to identify the enhancer sequences of a newly discovered gene. Shown below are the relevant regions of some of the reporter constructs the researcher designed to identify the enhancer. Now, what is this enhancer? Who can tell me what is enhancer? Is it a sequence? Is it a activator protein? Is it a transcription factor? What is an enhancer? What is its purpose? Let's see who can tell me this. I'm not asking for the answer of this question. I'm asking a very general question. Okay, a subject-based question. If you have studied transcription, you must be aware of what is enhancer. Okay, others. Others. This question is something which I ask in class also, because this concept is very confusing to many students. Okay, Maria is saying option 1. Chemistry says activator proteins enhance the process. Uh, Transcription factors binds anywhere, increase gene expression. Okay, enhancer is a transcription factor. Increase gene in case of splicing, it helps to load splicers of one. Okay, enhancer is a sequence. First and foremost, clear this idea right now, everyone. If this is your promoter site, this is the start point of your gene. Okay? Your gene is here. This is the promoter site or the core promoter where the polymerase is going to come and bind. What is this promoter? It is a sequence where RNA polymerase is going to bind assisted by the transcription factors. Transcription factors are proteins. RNA polymerase is an enzyme. ठीक है? But core promoter, when I say promoter, it is a sequence. You have some proximal control element under which you have the GC box and the cat box. These are also your sequences. We call it as proximal control elements. These are also known as your sequences. To this binds your activator proteins. Okay? Activator proteins binds here. What does it do? It it simply regulates the polymerase activity. Okay, it regulates the polymerase activity. Now, apart from this, so these all are present just next to your gene, upstream of the gene. Okay? Apart from them, you also have certain regions on the DNA which are known as your enhancer regions. These are also sequences. Enhancers are similar to enhancers are silencers. To the enhancer, transcription factors binds. Now these transcription factors are not same as the transcription factors here. Okay, these transcription factors are basal transcription factors. These are transcription factors, or many a times they are also known as activators. Now these factors or activators, when it binds to enhancer, they can activate the RNA polymerase. All right. Now there are certain characteristics of the enhancer. Enhancer is not necessary. It is going to be present just next to the gene. It can be far away from the gene also. All right. So similar to enhancer is silencer where repressors bind. So silencer is also a sequence. Repressor is a protein. Got it? So after this class, please open enhancer and read the characteristics of enhancer. Is very important. Many questions comes from enhancer. Okay? All right. Now the answer to this question is, one second. It is 3. Why? See, promoter is present before your reporter gene. Reporter gene means it is that gene which, when get transcribed, will give out certain signals that yes, transcription has happened. Okay, so if promoter is present before, please excuse me, if promoter is present upstream of the reporter gene, then obviously enhancer will be activating the polymerase sitting on the promoter, and this reporter gene will be transcribed. Okay, so A is correct. Now B. Here you only have the reporter gene, you don't have the promoter. Enhancers, the proteins which are sitting on the enhancers, or say the proximal control elements also, the activators basically, they cannot work directly on the gene. They regulate the polymerase for the transcription, to enhance the transcription or to activate the transcription. Okay, there are many regulations. I'm not going to the details of that. So if you don't have any promoter, where will the polymerase sit? So obviously this cannot be identified. Okay. Next you have the splice site acceptor. Okay. So yes, your enhancers also somehow regulates the proteins binding to the splice sites. Okay. So therefore, you will get some report from the reporter genes also. Okay. So answer 3 is correct. Now moving on to question number 9. So question number 9 is saying a guanine base in DNA is susceptible to modification to 8-oxo G because of oxidative damage. If modified guanine residues are not removed by a DNA glycosylase prior to replication, an adenine may be incorporated against G-oxoguanine. So just imagine there was a guanine against which you should have got a C, right? But this got modified to 8-oxoguanine. 8-oxoguanine structure is such that it will incorporate adenine as its complementary base. So there's a major change here. Okay. Enhancer is a sequence. Devashri. Enhancer is a sequence to which activators binds. Activators are proteins. ठीक है? Alright. To avoid mutations, the cells have another DNA glycosylase called MUT-Y. However, when the gene coding for MUT-Y is deleted, the strain survives. This observation suggests that in E. coli, what you have. So you have MUT-Y is not responsible for the excision of A incorporated against G-oxo, 8-oxo-G. Alternate DNA repair pathways may substitute for the repair of A. Now, before you come to the statement, have you understood the paragraph here? What is the story they are telling you? Yeah. So there is a change required. Okay, editing is required. There is this DNA glycosylase MUT-Y which can uh, edit this 8-oxo guanine. Okay, but in a cell where this MUT-Y is deleted, then there also we see that instead of the cell dying, the cell is surviving. That means what could have been possible? If this exists, the cell will die. Who can repair this? MUT-Y. Right? Now we find that MUT-Y is also mutated, not present. Still, the cell is surviving. That means there must be some alternate DNA repair pathway which has substituted for the repair of A incorporated against 8-oxo guanosine. Okay? So this got incorporated, but this adenine got substituted by other repair mechanism. As we know that there are more than one repair mechanism existing in the cell. Okay? Now the last question. Very interesting question and very easy question. But you know what? Most of the students, they will get scared by just looking at this question. Why? Because there's a lot of graphs and everything given here. No point getting scared. Just read it once. You will see that it is very easy. They are telling you that a construct was generated to access the activity of bidirectional promoter. Okay, so this is your promoter A, this is your promoter B. Okay, and against each of the promoter there is a reporter gene given here. So if this promoter gets activated, reporter gene 1 will be active. If this gets activated, promoter B, then you will get some expression from gene 2. That is how the construct has been made, which has a common regulatory DNA element X. The construct was used to transform E. coli. The activity of the promoter is recorded in the presence of increasing level of an E. coli encoded regulatory protein. Okay, so this is the regulatory protein which regulates both these promoters. Okay? Increasing level of an E. coli encoded regulatory protein which binds to DNA element X. Okay, so this is an element where the E. coli regulatory protein comes and binds, and it has got some impact on the two promoters. That is the meaning. Okay, so this is what you get to observe. This is x-axis is showing you increasing level of regulatory proteins and activity of the reporter gene. Okay? So as we see here, that as your reporter gene levels are increasing, gene 1 activity is getting reduced and gene 2 activity is increasing. This is a standard. Okay? Now, the above experiment is repeated in a mutant E. coli with mutation which abolishes the binding of the regulatory protein to element X. That means a mutation has happened this site, at this site, therefore, the regulatory protein cannot bind here. Alright? Which one of the following graphs best depicts the activity of the reporter genes in the mutant strain? So, option 4. Why option 4? This is correct. Dekho, look at this graph. Initially, the reporter gene 1 expression was very high. You know? And then, under the influence of this regulatory protein, the expression came down. And initially, the reporter gene 2 expression was very low. Under the influence, it came up. So, if there is no influence, then this modification will not happen. You know? So, the graph will be absolutely straight. Gene 1 expression will keep being high, and gene 2 expression will be maintained in low condition. Agree, everyone? Okay? So, this is very logic. There is nothing theory here, present here. A very simple logic you have to apply. And that's all from my end today. If you want to practice more questions, download Biotecnika's app. It is free. And there you will get this T3 test series. Okay, every day we are uploading some tough net questions. Okay, 10 questions we are uploading. Practice answering the questions because this is what you can do best just before your exams. Your exams are around the corner. And those who want to study with us, get in touch with us. We have this lecture batch. All right, we have unlimited classes, chat supports, dedicated study portals. Conceptica series comes free. Test series, obviously, there are many test series we have. It also comes free. So, if you want to know in details, contact in this toll free number. We will be happy to help you out. With that, thank you, everyone, and wish you all the best. Keep revising, keep practicing. Meet you in the next live class.